What I would like to do during this talk is to highlight one or two areas where I think RRI has in the past played an important role, and ones that it's going to play an important role in the future. And I will introduce a few different topics along the way. Now, I'm going to start off by talking about genetic engineering and synthetic biology. And I was involved in this in the very early days. I attended the Asilomar conference in 1975, which was one of the first real attempts by the molecular biology community to behave responsibly. And this was a conference organized by Paul Berg and several others. And there were two interesting features that came out of that. One was that there was a great deal of concern among scientists in the molecular biology community about what they were doing and potential dangers. But after the conference and after a number of people had spoken, it was clear that most molecular biologists knew much less about real biology of bacteria than people who'd worked in the field for a long time. And so it was a case where some experts couldn't really agree at the time on what was the correct path forward. As a result of the Asilomar conference, cities like Cambridge in Massachusetts put in place orders that stopped people from doing recombinant DNA technology. And it wasn't until many years later that a number of these restrictions were stopped and people were allowed to make recombinant bacteria without too many structures. But even today, we find that there are restrictions on just what you can and can't do when you're doing recombinant DNA. In bacteria, it's been fairly loose, I would say. And part of the reason is because you can contain any bacteria you make, and so it is easy to behave in a fairly responsible manner. However, when we come to plants and the possibility of changing plants using genetic engineering or synthetic biology, this has become much more of a contentious issue. But I would argue that it's become a contentious issue because of money and because of politics. A great deal of what went on in Europe in anti-GMO movements was based upon anti-Monsanto movements more than anything else, and a desire by the Green parties to try to stop the US from taking over the food supply in Europe. At least this was the underlying argument that was made. And Greenpeace leapt on this and got a very highly organized political campaign going as a result of it. They claimed at the time, and they've always claimed, that science is very important to them when making decisions. But in the case of GM plants and GM crops, the science is really very clear. At the present time, after some 30 years of experience, there's not been a single problem. And yet Greenpeace continues to be opposed to this. I would argue strongly that responsible research in the plant field demands that we use the very best techniques available, and genetic modification is one of the best techniques, to enable the developing countries to produce the food that they need. We in the developed world should not be telling them how they should be improving their crops and feeding their people. For me, this is a moral issue that I find somewhat repugnant by the attitude of people who would try to stop people in the developing countries improving their crops. Now, another area, of course, are animals. We've done a certain amount of genetic modification of animals. Pigs, for instance, we've made humanized pigs because there you can get organs which are suitable for research, to try to study how, maybe for transplantation, or to study how the organisms are working. And of course, animals we've been genetically modifying for years by somewhat traditional means. For instance, most of the dogs that we have as pets can scarcely be considered natural anymore. They've been very heavily bred and modified over the years. I have no particular problems with modifying animals for agricultural purposes, or even as pets. But at the moment, I don't see that it's necessary to do more than a small amount of genetic engineering for that purpose. Finally, we come on to the issue of humans, which is probably the most contentious issue just at the moment, as to whether or not we should be making modification of humans. The recent experiments that were carried out in China by Dr. He really raised some very serious ethical questions. And my own view on modification of humans, particularly the germline, while for the rest of the body, for say replacing blood to solve sickle cell anemia, that kind of thing, I think this is okay. I don't have a problem with that, although obviously one would like to put in place the very best guidelines for doing such work. But when it comes to germline editing, I think we have to be very clear. One, we have to make sure that it's done in a very safe manner. We have to make sure it's only done for purposes that could not be done by any other means. And so, for instance, eliminating genes that lead to severe genetic deformities is an area that I would consider possible. The experiments that were carried out by Dr. He were really unethical, in my view, in every conceivable way. But we need to know that the methods we're going to use, and in this case, CRISPR technology is the one that everybody is using at the moment, these need to be done in a way that we know that the targeting is very precise. We don't want to see side effects that might be a problem. And I think CRISPR technology has not yet reached that point, in my view. And maybe later on, other technologies will come along that are better, and we should then judge them at the time as to whether or not this is suitable. I also think that we need to take into account the possibility for side effects that we had not considered when we're doing these kinds of experiments. So, for instance, we know very well that if the BRCA gene is present, that this leads to an increased likelihood of breast cancer for ladies. Now, we know that there are many people who have a defective BRCA gene who never get cancer. And the answer is we don't know why they don't get cancer. And it could well be that there are compensatory mutations that actually are keeping such people alive successfully, and that could be present for many other diseases too. So, for instance, people who have the Huntington's gene usually get Huntington's syndrome if they live long enough, but apparently I'm told there are one or two who don't, and we don't know why. There are many other genetic diseases of that sort where you can get the disease, you can have if you have the gene, but you can also not get the disease if you have the gene. And so the interactions that take place within various genes in the body are not well understood at the moment. Now, this leads on to the next topic, which is the topic of DNA sequencing. What's happened over the last 10, 20 years is that we've become very good at sequencing DNA. It is now quite easy to get a complete human genome sequence. The cost is not prohibitive. In fact, when we're starting to study biology, no matter what biology, usually one of the first things we do is to sequence the organism that we're interested in and use that sequence as the basis for understanding the biology of the organism. In fact, it's becoming clear that over the course of the next 50 years or so, we're going to sequence everything that is present, living on Earth. We're already sequencing all the bacteria that live with us. We're sequencing animals that are out in the wild. I think this, on the whole, is a good thing, but only if it is followed by a serious attempt to put the sequence knowledge into the context of the biology. That is, if we sequence something, we find some genes, we need to find out what those genes are doing. And I also believe that contrary to what is happening in some countries at the moment, we should be sharing those DNA sequences. I've always been a big believer in open access for science and for open access for both communication and for DNA sequence information. I was instrumental in getting GenBank started, which was the first DNA database that was publicly available to everybody for sequence information. And I follow very carefully the progress of GenBank and EBI and other databases, the DNA database of Japan and so on. But in China, there is a lot of sequencing taking place, and that data is not being shared. Now, of course, when it comes to humans and human sequences, there are very strict ethical procedures that need to be in place before we begin to share human DNA sequence. First of all, I think every individual has the right to keep their sequence private if they want to do so. I don't think this is something that politicians or anybody else should be deciding. I think this should be a very personal view as to what is being kept private. Now, you can argue that, say, for forensic purposes, and this is not necessarily the case, perhaps we shouldn't allow everybody to keep it private. And that, I think, is a debate that is worth having. It's something that's worth talking about. Perhaps if certain definitive characters, parts of the sequence were made available, that's okay. But again, I think this is a debate that needs to take place between criminologists, between scientists, between the general public, and between ethicists. This is a very difficult situation to deal with. On the whole, I personally would be happy to have my sequence made available to anybody who wants it, and I think there are many such people. And this whole idea of individual responsibility leads me to the last point that I want to make, and that relates to coronavirus. We're all very concerned as scientists at the moment about coronavirus, and I think the scientific community has been exemplary in mobilizing to a very large extent, looking for ways to provide better tests, to provide therapeutics of one sort and another, to provide vaccines, and so on. There's been a tremendous amount of activity. People who would never have ever dreamt about talking or working on coronavirus have stepped up. There is a great deal of talk at the moment about a vaccine. Obviously, if we had a vaccine, that is going to make life much easier for those people who take the vaccine, have proof that they've taken it, and it works, and then they can resume their normal activities. Things like flying, things like going back to work, all of these would benefit greatly if a good vaccine was available. Now, the way we traditionally make vaccines is actually a rather slow process. But I would argue, and argue quite strongly, that it needn't be. There has been tremendous ethical backpush against the idea of testing a vaccine on human volunteers. And by testing on human volunteers, I mean to deliver the vaccine to them, let the vaccine work and produce antibodies for a while, and then to deliberately infect them with the virus to see if the vaccine works. I'm told that in the UK, more than 3,000 volunteers have already stepped forward, ready to do this, should it become possible. I believe that in the case of coronavirus, which is devastating the world, this is a very good way to test early vaccines. If we select people who are in an age group where it's unlikely that they would die if they got the virus, I think if they then choose to volunteer to be test subjects, I would heartily approve of that. And I think this is something that perhaps for the first time in the vaccine world, we could think about doing some far-reaching science that may have tremendous consequences going forward. It may allow us to develop vaccines far more rapidly than we have in the past. And I will just close by saying that I have no sympathy at all for the people who don't want to be vaccinated against anything. I think they've been fed a tremendous amount of false information. And I think we need to find a way in science of distinguishing truth from lies in ways that we've not been very good at doing in the past. And one way we should do that is by making sure that scientists speak out to the general public and speaking language that the general public can understand. I think we as scientists have fallen short in that regard, and it would be better if we could, in fact, speak clearly to the general public. I would make this compulsory education for all scientists. And on that note, I'll close, and I'm happy to take any questions.